In this video, we are going to talk about DNA fingerprinting. All right, before we get started, we do need to go over some important people in DNA history. You already know about Watson and Crick from biology class. They are the ones that came up with the double helix shape for the DNA molecule. Then Alec Jeffreys and Kary Mullis, they're both from England, and Jeffreys, he is the biggie when it comes to DNA. In 1985, he published that he had been able to isolate specific markers that made DNA unique to each individual person, and he called them DNA fingerprints. Kary Mullis was working at the same time with DNA, and he came up with something called PCR, and we will talk about that in a minute. You do not need to know the dates, but it is kind of interesting to know how far we've come in such a short period of time. In 1988, the FBI started using DNA in their casework. 1998, they came up with the CODIS database, and so that's not all that long ago. DNA has come really far in just a very short period of time. Where is DNA found? You actually know the answer to that question already. We have talked about any cell that has a nucleus will have DNA in it. Remember, red blood cells do not have a nucleus, so they do not have nuclear DNA in them. Here's the basics of DNA. You guys should already know that it is a double helix. It's two strands of a bunch of nucleotide bases. Their specific pairing, and the way I remember it is A has straight sides to it, T has straight sides to the letter, and so A and T go together. C is a curved letter, G is a curved letter, and so they go together. That's just the way I remember it. For humans, we actually match about 99.9% of the time. It's that 0.1% that makes us unique. DNA typing is the method in which we are able to create somebody's DNA fingerprint. It's a series of bands that is unique to one specific individual or identical twins. As we said on the previous slide, only a very small part of us is different from one another. Jeffreys came up with the way to focus on that small part that is different and be able to pinpoint it and be able to create that fingerprint that will let us tell the difference from one person to the next. Here are some uses for using DNA. It sometimes is called DNA profiling. We'll use them to identify suspects, to exonerate somebody like the Innocence Project does, to identify victims, to determine maternity or paternity, who's the baby daddy, to deal with organ donation. There are some parts of DNA that help make organ donation a better match. And any other kind of human remains, not necessarily part of a crime or anything. All right, when we are collecting anything that might have DNA on it, there are very specific ways that we need to collect. We need to avoid any kind of contamination. First of all, everything should be disposable that we use. It just makes life easier. You need to not talk, breathe, sneeze, cough, anything over the evidence area. Lots of places will require their technicians to wear masks. Anytime you talk or breathe, little bits of spit go flying out. You can't really see it, and DNA is in there, and you don't want your DNA being on the DNA that's found at the crime scene. We've already talked about point number 3 on here. You need to make sure that all the evidence is put in something that can air dry. It should not be airtight. You want air to be able to dry it out. If it cannot be dried, they'll freeze it. A lot of really big forensics labs will have these huge freezers for like mattresses that are soaked in blood. They'll go ahead and they'll freeze it instead of trying to dry it out. And the evidence should be cool and dry whenever you're transporting it or storing it. It will make the DNA be preserved much better. Sometimes you'll hear something referred to as a standard or a reference, and those are DNA samples that are collected from other people. So let's say somebody is murdered in our class and they think that the suspect is somebody in the classroom. They would go and swab all of us to get standards. They would have our names written on the swabs. They would know who belonged to that. And then they could take the sample that is found on the victim and they could test it and see who it matches with. They don't always take blood anymore. Lots of times they take what are called buccal cell swabs. That is your cheek cells, and it's real easy to do that. Not painful at all. All right, we are going to go through the steps of processing DNA. Step number 1 is extraction. So you've swabbed somebody's cheek, you've gotten cheek cells. You need to get them off of the swab and isolate the DNA. So they clean it, there's detergents that are used, and it will separate the DNA from everything else. Step number 2, they are going to use restriction enzymes to cut it in specific places. Here are a couple examples on the screen. There are many different types of enzymes, and they will cut in different ways. They're looking for different patterns on the DNA, and they will cut it in different places. We will look at this a little bit more in class, but it's kind of cool the way it can cut. It knows, like in the one pattern, every time it sees CCC, GGG, it will cut right in between the C's and the G's. It won't cut anywhere else. This will help them deal with smaller pieces. Step number 3 is called amplification, and this is where Kary Mullis comes into play. He came up with this procedure of using PCR. PCR is a kind of complicated method, but you do not have to know exactly how it works. But what it does is it amplifies a sample. Amplification just means to copy it. Amplification, though, is the right word. So what it does is you have a little bit of DNA found at the crime scene. Then they'll put it in the machine to do PCR. The PCR will go through multiple cycles, and it will keep copying the DNA over and over and over again, and you'll end up with a lot of DNA to be able to test. All right, then the next step is gel electrophoresis. Gel electrophoresis will separate all those different fragments that you have from cutting it all up. It will separate it all apart and allow you to get the bands. What they have to do is they get these gels. It's kind of like a really thick jello, and they'll load the DNA in there. DNA has a charge to it, and so if you put electricity through, you can make the DNA move. So here's a little picture here. DNA has a negative charge, and so it's going to be attracted to the positive end of the little box that you deal with, and the little pieces of DNA will move through the gel, and you end up having DNA at different spots. The shorter the piece, the further it will move, and so it will really separate it out. Again, we'll look at this in class, but it's kind of neat what they're able to do. Here's another picture that shows DNA samples and the different bands that they can have. Lots of times you have to stain it in order to see it. Blue is a very common color. The last step on here is to analyze your results. Depends on what process you are doing as to what you need to do. But one process is called southern blotting. Has nothing to do with the South. It's somebody's last name. And what they are able to do is they actually put radioactive material in with the DNA sample, and it will attach to certain fragments of DNA, and then when you put an X-ray on top of it, it will develop the X-ray and you get these bands. This is a picture of southern blotting. And you can analyze all of them. This is just kind of a cartoony, cutesy picture. Here are some real samples. The latest method of DNA typing is something called short tandem repeats, STR. What this is they have figured out how to use really, really small pieces of DNA to create a DNA fingerprint. That means that if DNA starts to break down, like old cases from the Innocence Project, and the DNA is broken down a little bit, it's okay. It uses these really short sections on our DNA that repeat themselves. And so it's called STR, and its benefits are they can use much smaller samples. That's the key thing you should know about this. CODIS is a database that stores all the DNA profiles that have been developed from convicted offenders, unsolved crime scene evidence, profiles of missing people. Anytime DNA is processed, it can get put into CODIS. Right now, not everybody's DNA is in there. There are people that want that to happen, but I don't see that happening anytime soon. All right, just another quick review here of mitochondrial DNA. We've talked about this before. This is the DNA that comes from your mitochondria, not from the nucleus of a cell. And so it is the one that ties specifically to your mother's side of the family. It only uses little tiny bits of DNA. It can be just from hair, not the follicle cells, but just from the hair itself. But it's really expensive to do, and so they tend to only do it if they really have to.